So, have you ever wondered how blood is formed in our body? Because people lose blood very often, and we may wonder how is it restored. So, let's check it out. The process of formation of blood cells in our body is known as erythropoiesis. So in this video, we're going to enumerate the definition, the site of blood formation, that is in fetal life, newborns, children, and adults, the process of blood formation, and also the factors which affects the formation of red blood cells in our body. Definition. Erythropoiesis is the process of origin, development, and maturation of erythrocytes, that is, the RBCs. Site. In fetal life, the erythropoiesis occurs in three stages. Number 1. The mesoblastic stage, that is, during the first 2 months of intrauterine life, the RBCs are produced from mesenchyme of yolk sac. Number 2. Hepatic stage. From the word itself, we can identify that it's something related with the liver. So from the 3rd month of intrauterine life, liver is the main organ that produces RBC. Spleen and lymph organs are also involved in erythropoiesis. Number 3. Myeloid stage. During the last 3 months of intrauterine life, the RBCs are produced from the bone marrow and liver. In newborns, children, and adults, the RBCs are only produced from bone marrows. Up to age of 20 years, RBCs are produced from red bone marrow of all bones. After the age of 20 years, RBCs are produced from membranous bones like vertebra, sternum, ribs, scapula, iliac bones, and long bones. Because of fat deposition, shaft of long bones become yellow and loses the erythropoietic function. But in adults, even if bone marrow is destroyed, liver and spleen produces blood. Bone marrow only involves 25% of RBC production. The rest 75% is involved in the production of leukocytes, that is WBCs. Still, leukocytes are less in number because of lifespan of leukocytes. Lifespan of erythrocyte is 120 days, whereas that of leukocyte is 1 to 10 days. Process of erythropoiesis. Stem cells. These are the primary cells capable of self-renewal and differentiating into specialized cells. Hematopoietic stem cells are the primitive cells in the bone marrow, which give rise to the blood cells. Hematopoietic stem cells in the bone marrow are uncommitted pluripotent hematopoietic stem cells. In short, we call them PHSC. PHSC is defined as a cell that can give rise to all type of blood cells. In early stages, the PHSC are not designed to form a particular type of blood cell, and it is not possible to determine the blood cell to be developed from these cells. Hence, the name uncommitted PHSC. In adults, only a few of these cells are present. Best source is umbilical cord blood. When the cells are designed to form a particular type of blood cell, the uncommitted PHSCs are called committed PHSCs. Committed PHSC is defined as a cell which is restricted to give rise to one group of blood cells. Committed PHSCs are of two types. Number 1, lymphoid stem cells, which give rise to lymphocytes and natural killer cells. Number 2, colony-forming blastocytes, which give rise to myeloid cells. What is myeloid cells? These are the blood cells other than lymphocytes. When grown in cultures, these cells form colonies, hence the name colony-forming blastocytes. Different units of colony-forming cells are: number 1, colony-forming unit erythrocytes, in short we call them CFUE. Cells of this unit develop into erythrocytes. Number 2, colony-forming unit granulocytes or monocytes. These cells give rise to granulocytes. The granulocytes are neutrophils, basophils, and eosinophils, and monocytes comes under the agranular division of the WBCs. colony-forming unit megakaryocytes. In short, we call them CFUM. Platelets are developed from these cells. So let's move on to checking what are the changes during erythropoiesis. Cells of CFU erythrocytes pass through different stages and finally become the matured RBC. Four important changes are noticed. They are: number 1, reduction in size of the cell; number 2, disappearance of nucleoli and nucleus; number 3, appearance of hemoglobin; number 4, change in the staining properties of the cytoplasm. Let's check the stages of erythropoiesis. So various stages between CFU erythrocyte cells and mature RBCs are: number 1, Proerythroblast; number 2, early normoblast; number 3, intermediate normoblast; number 4, late normoblast; number 5, reticulocyte; number 6, matured erythrocyte. Now let's go in detail. Number 1, Proerythroblast. Proerythroblast or megaloblast is the first cell derived from CFUE, that is erythrocyte. It is very large in size with a diameter of about 20 microns. The nucleus is large and occupies the cell almost completely. The nucleus has 2 or more nucleoli. Proerythroblast does not contain hemoglobin. The cytoplasm is basophilic in nature. Proerythroblast multiplies several times and finally forms the cells of next stage, which is early normoblast. Synthesis of hemoglobin starts at this stage. But the appearance of hemoglobin occurs only at the intermediate normoblast. Moving on to next, the early normoblast. This cell is a little smaller than the proerythroblast with a diameter of 15 microns. In the nucleus, the nucleoli disappear. Condensation of chromatin network occurs. The condensed network becomes dense. Cytoplasm is basilic in nature, so the cell is also called basophilic erythroblast. This cell develops into next stage called intermediate normoblast. Intermediate normoblast, the cell is smaller than early normoblast with diameter of 10 to 12 microns. Nucleus is still present, but chromatin network shows condensation. Hemoglobin starts appearing. Cytoplasm is already basophilic. Because of the presence of hemoglobin, it stains with both acidic as well as basic stains. So this cell is called polychromophilic or polychromatic erythroblast. Cells develop into next stage called late normoblast. For late normoblast, diameter of cells decreases further to about 8 to 10 microns. Nucleus becomes very small with very much condensed chromatin network and is also known as ink spot nucleus. The quantity of hemoglobin increases. Cytoplasm becomes almost acidophilic. In the final stage of late normoblast, just before it passes to the next stage, the nucleus disintegrates and disappears. Process by which nucleus disappears is called pycnosis. Late normoblast develops into reticulocyte. Reticulocyte is otherwise known as immature RBC. It is slightly larger than mature RBC. The cytoplasm contains the reticular network or reticulum. Due to the reticular network, the cell is called reticulocyte. In counts, in newborn babies, the reticulocyte count is 2% to 6% of RBC. That is, 2 to 6 reticulocytes are present for every 100 RBCs. The number of reticulocytes decreases during the first week after birth. Later, the reticulocyte count remains constant or below 1% of RBCs. The number increases whenever production and increase of RBCs increase. Reticulocyte is basophilic. Matured erythrocyte or RBC. For this, the reticular network disappears. The cell becomes the matured RBC. It attains the biconcave shape. The cell decreases in size. The matured RBC is with hemoglobin but without nucleus. But for the camels, there is an exception. The camels possess RBCs with nucleus. So, coming back, it requires 7 days for the development and maturation of RBC from proerythroblast. It requires 5 days up to the stage of reticulocyte, and reticulocyte takes 2 more days to become a matured RBC. So now let's move on to the factors necessary for erythropoiesis. Development and maturation of erythrocytes require a variety of factors, which are classified into 3 categories. Number 1, general factors. Number 2, maturation factors. Number 3, factors necessary for hemoglobin formation. General factors. General factors necessary for erythropoiesis are: number 1, erythropoietin. Number 2, thyroxine. Number 3, vitamins. Now let's check in detail. Erythropoietin. It is the most important general factor for erythropoiesis. It is also called hematopoietin or erythrocyte stimulating factor. Chemistry. Erythropoietin is a glycoprotein with 165 amino acids. Source of secretion. Major quantity of erythropoietin is secreted by the peritubular capillaries of kidney. A small quantity is also secreted from brain as well as the liver. Stimulant for secretion. Hypoxia or decreased availability of oxygen is the stimulant for the secretion of erythropoietin. Actions of erythropoietin. Erythropoietin causes formation and release of the new RBC into circulation. After secretion, it takes 4 to 5 days to show the action. Erythropoietin promotes the following processes. A. Production of proerythroblast from CFU erythrocytes of the bone marrow. B. Development of proerythroblast into matured RBCs. C. Release of matured erythrocyte into blood. Number 2 is thyroxine. Being a general metabolic hormone, thyroxine accelerates the process of erythropoiesis at many levels. So, hyperthyroidism and polycythemia are common. 3. Vitamins. Deficiency of these vitamins cause anemia associated with other disorders, which are vitamin B. Its deficiency causes anemia and pellagra. Vitamin C. Its deficiency causes anemia and scurvy. Vitamin D. Its deficiency causes anemia and rickets. Vitamin E. Its deficiency leads to anemia and malnutrition. Maturation factors. Vitamin B12, intrinsic factor, and folic acid are necessary for the maturation of RBCs. Vitamin B12 is also known as cyanocobalamin. It is the maturation factor necessary for erythropoiesis. Source. Vitamin B12 is called extrinsic factor since it is obtained mostly from diet. Vitamin B12 is stored mostly in liver and in small quantity in muscle. It is also produced in large intestine by the intestinal flora. Action. Vitamin B12 is essential for the synthesis of DNA in RBCs. Its deficiency leads to failure in maturation of the cell. Deficiency of vitamin B12 causes pernicious anemia. Number 2. Intrinsic factor of Castle. Intrinsic factor of Castle is produced in gastric mucosa by the parietal cells. It is essential for the absorption of vitamin B12 from intestine. In the absence of intrinsic factor, vitamin B12 is not absorbed from intestine. This leads to pernicious anemia. Deficiency of intrinsic factor occurs in severe gastritis, ulcer, gastrectomy. Number 3. Folic acid. Folic acid is also essential for the maturation. It is required for the synthesis of DNA. The absence of folic acid leads to anemia in which the cells are larger and appear in megaloblastic stage. Anemia due to folic acid deficiency is called megaloblastic anemia. Factors necessary for hemoglobin formation. Various materials are essential for the formation of hemoglobin in the RBCs. Deficiency of these substances decrease the production of hemoglobin, leading to anemia. Such factors are number 1, first-class proteins and amino acids. Number 2, iron, which is necessary for the formation of heme part of the hemoglobin. Number 3, copper, necessary for the absorption of iron from the gastrointestinal tract. Number 4, cobalt and nickel. These metals are essential for the utilization of iron during hemoglobin formation. Number 5, vitamins. Vitamin C, riboflavin, nicotinic acid, and pyridoxine are also essential for the formation of hemoglobin. Hope you have understood the video. Like, subscribe, and press the bell button for more videos. Thank you. Thanks for watching.